sounds like we've got a critical mass now, and it's recording anyway, so might kind of get the ball rolling a bit. So normally I try to make these tutes as interactive as possible, which obviously has its challenges online, but there are some aspects that make it easier when compared to having a couple of people in a tute room and everyone else video conferencing. Zoom can almost kind of work better with this in terms of sound quality and muting and those sorts of issues. So we'll see how we go, but yes, I'm more than happy for people to interrupt me, stop me, unmute yourself, or whether you want to raise your hand or put stuff in the comments. Maybe Ben, you can keep an eye on the comments and let me know if there's specific questions. But as I said, I'm more than happy for you to interrupt me, and I'll probably pause at various stations asking questions and waiting for answers, as well as giving people to ask questions of their own. I pretty much give the same tute every year, and every year I tell myself, oh, I'll just kind of redo what I did last year, but it never ends up happening. So yet again, I've kind of tweaked things and put new stuff in and changed my slides around. So it's could or turn into a mess when I kind of get wrong slides or forget the new bits or whatever, but we'll see how we go. That's one reason I like to put my tutes, including the slides, online, because they do change a bit from year to year. So if people want to kind of spend a couple of hours listening to me talk on this topic from last year, you're welcome to do so. And yes, the link to my primary resources on my blog site where I store those files is in the chat already. Anyway, I'll start sharing my screen. And yes, at one point I'll be trying to share from my iPad as well, which could make everything fall in a heap, but oops, that didn't quite work. Okay, so is that working, everyone? You can see my slide. Yep. We can. Yep, good. Okay, so yes, first up, does anyone want to have a crack at what this is a picture of, an electron micrograph?
I don't know if it's a neuromuscular junction.
Well, you're right, Daniel, it is. Nice work, and yes, possibly the title of this week's tute was a bit of a giveaway in that respect. So yes, that is a neuromuscular junction. So the kind of the bright white bit is the neuron, and then it's finishing on the muscle cells down there with that can see the synapses and everything. Yes, and so our topic for today is nervous system and muscle physiology. And yes, those of you that I haven't met or don't know, my name's Rhys Thomas. And yes, I sat the primary way back in 2008, which is getting longer and longer ago every time I give these tutes. And there's another link to the blog site where I keep some primary exam resources, and that link is also in the Zoom chat, so you're more than welcome to make use of them. And I'm keen for feedback if you think that there's stuff that's in there that's kind of unnecessary or new stuff has crept in, because yes, the syllabus has obviously changed since I sat it, and these tutes are kind of based primarily on the old syllabus, which I'll kind of get to on the next slide. So yes, this is, I'll kind of do this with each of my tutes. So this is how they laid out the learning objectives in the primary syllabus when I sat it. There was a whole kind of file that had for each topic what the learning objectives were. They were kind of laid out, and you could go through and tick each one to make sure that you'd covered it in some degree. You had to make a judgment call in terms of what was core and what was more peripheral. But it was, a very helpful way to organize it. Whereas I think with the change in the syllabus, it's now kind of spread out a bit more and a bit more nebulous and harder to find. So on my blog site, there's a link to the PDF that has all the different topics in it. Now, as I'm sure you know, there are some things that are no longer in the primary syllabus and other things have been added in. So it's obviously not the be all and end all, but it might still be helpful for you to make sure that you cover most of the topics as they were back then anyway. So yes, that's the nervous system physiology bit. That's the muscle physiology, just for reference. And for starters, I'll kind of do my usual disclaimer about spherical cows. If you've been to my other tutes, you're probably sick of this joke by now, but the concept of a spherical cow came about from this joke. And so a farmer employs some scientists and engineers to work out the most efficient way to milk cows. And after lots of time, effort, and money, they eventually come back to him with their solution. And it goes something along the lines of, well, let's consider a perfectly elastic spherical cow in a vacuum. And so the idea behind the concept of a spherical cow is that often to be able to understand or explain things, we have to grossly simplify them to the point where verging on just being wrong or misleading. But I do think that a lot of these spherical cows can be very helpful to get an initial understanding of some concepts. And there's always the opportunity for you to go down rabbit holes or dig a bit deeper. But yes, it's also just a warning that if you do start digging a bit deeper, you will find things that don't quite match up with what you've been taught in lectures or textbooks, and it will differ between textbooks as well. But one of the important things is that I think, from my understanding anyway, when I sat it, was that most of the examiners kind of, they're happy with the spherical cow explanations in terms of getting through the primary exam. So for this particular topic today, we'll start off with just a bit of microanatomy. So yes, this is a stylized neuron. Now, does anyone want to kind of go through what the different bits are? So can you see my mouse pointer okay? Yep. Okay, so does anyone want to, yes, what's this line pointing to here?
The nucleus?
Nucleus, yep, good. What about, yeah, this one's, yeah, let's go with this one.
The axon hillock?
That's, yes, well, yeah, we'll certainly get to that topic, but I think it's just more generically talking about the soma, the cell body. So yes, we'll certainly come to the axon hillock. I don't think it's explicitly indicated on this one. What about this black line up here? What's it pointing to?
The dendrite.
Dendrite, yep. This one down here.
Myelin sheath.
Yes, and what produces the myelin?
The Schwann cell, if
yep.
it's CNS, or oligodendrocytes, if it's CNS.
Oh, yes, very good. This one, I'm assuming, or maybe, yeah, maybe this one's pointing to the myelin and this one's pointing to the Schwann cell, perhaps, or at least its nucleus. What about this one here?
The node of Ranvier.
Yep, and then finally,
the synaptic cleft.
Yeah. Oh, well, yes. I guess there's yes. So the synapse, or possibly sometimes they talk about the end of the axon as being a terminal bouton or something in French. But yes, that's so that's a pretty simplified spherical cow of a neuron to get the ball rolling. So now a lot of the research and study, historically speaking, looking at neuronal physiology and the electrochemistry of it, started by looking at the giant axon of the squid. And I remember when I was doing physiology as an undergraduate, and they talked about this giant. This is what I was picturing. It was an axon from a giant squid. But it's not actually quite like that. It's so every squid has a giant axon, and I'm sure the giant axon or the giant squid would be even bigger. But yes, there's these particular axons in squids that are big, and so apparently they're up to 1 millimeter in diameter. So theoretically visible with the naked eye even. Whereas, yeah, most of the stuff we deal with in this sort of stuff is kind of microscopic, if not electron microscopic. So this really is quite a large axon in this context, and that made it relatively easy for them to study. And yes, one of the core concepts that they used these giant axons to study was the concept of a resting membrane potential. And I covered this a bit in my earlier shoot on cellular physiology, and this is a bit of a spherical cow in terms of how we describe it, but it is kind of a pretty fundamental understanding as to how kind of excitable cells work. So yes, is anyone willing to have a crack at describing, well, yeah, first of all, what is the resting membrane potential, and then we'll go on to how it's generated or maintained. So anyone willing to start throwing out some phrases or descriptions? No one's game?
I can give it a crack, but if other people want to have a go, I'll take a back seat.
Now I can go for it, Daniel. Oh, I think you're muted, Daniel.
The resting membrane potential refers to the potential difference in electricity on either side of the membrane, with it being more negative inside than outside.
Mm-hmm.
And this is due to the Nernst potential of different electrolytes that have differing degrees of permeability through the cell.
Yep.
So
okay.
The primary factor is that you'll have a high concentration of potassium that wants to spill out of the cell, and it will do so until it hits the equilibrium of its electrical gradient, balancing in versus out, and that's -90.
Right. Okay. Yeah. So yeah, no, that's a pretty good description of it. So I guess going back a couple of steps, you mentioned kind of the Nernst potential there, and kind of these gradients. So yeah, what is it that generates this gradient for potassium, and what else is affected by that?
So my understanding is that it is the impermeability of the cell membrane to sodium and chloride, but permeable to potassium. Yep. You've got lots of negatively charged proteins inside the cell, so a positive cation is going to accumulate inside. It will spill out, and then that gradient will be maintained only to a minor degree by the pumping the sodium potassium ATPase, which pumps out 3 sodium and in 2 potassium.
Yep. Yes. So yeah, you're certainly on the right track there. I guess yes. Just you mentioned there that the gradient is partly maintained by the pump. I think the pump is the main driver of the gradient, but it has a small contribution to the membrane potential. Okay. So yeah, and so yeah, you kind of talk about they're soluble to potassium, but not so soluble to those other things. And so yeah, I guess the buzzword there, if you're trying to get it out in a Viva or a written question, is that's kind of referred to as a semi-permeable membrane. So it's permeable to some things, but not others. And yes, you pointed out there that it has some background permeability to potassium. So that's one of the key words to get out in the context of the resting membrane potential. Yes, then the other one is these concentration gradients, and it's important that you know how these concentration gradients develop. And yes, as you said, that's through the sodium potassium ATPase. So that pumps sodium out of the cell and potassium in. So you get this different concentration, or it's a high concentration of potassium inside the cell relative to outside, and the reverse for sodium. And you mentioned those gradients, and then there'll be both a chemical gradient for those ions as well as an electrical gradient. And then you mentioned the Nernst potential that will determine where that settles down. And yes, as you said, it's more negative inside than outside of the cell, relatively speaking. And it's important to remember that the resting membrane potential is a bit of a misnomer. As we've said, it's generated and maintained by the sodium-potassium ATPase, which is a very active process. So even to get into this so-called resting state, it's a dynamic equilibrium because there's constantly potassium leaking out, and the sodium-potassium ATPase is constantly having to pump things back in again to maintain those concentration gradients. And it'll, the resting membrane potential will differ significantly between different cells. And the idea of it being resting is mostly in the context of excitable cells, which is kind of the focus of this tute. Just having a look through my notes here. Yeah, so this next slide, I think this is from Ganong, but I think he's pinched it from somewhere else. So this kind of highlights, yeah, kind of the membrane potential, but it also highlights the importance that this this potential difference is really only of significance right at the edge of the cellular membrane. Like, it's not that the whole of the inside of the cell is negative and the outside is positive. By and large, they're electroneutral, but just lined up along the very edges of the plasma membrane is this potential difference across it. So in terms of the actual movement of those ions, the actual net amount of ionic movement is relatively small compared to all the charges within and without the cells. So that's an important thing to remember. Any questions or clarifications about this at this point, or anyone else want to throw in anything before we move on? No? Okay. Yes, well, yes, you kind of started touching on this before. So, yes, does anyone know what this this Goldman equation is? And it's kind of, yeah, related to the Nernst potentials as mentioned before. Who wants to have a talk through this?
So it's a calculation for the contribution of the three major ions and how they affect the membrane potential. So in the numerator, you got the outside potassium, outside sodium, and inside chloride, and then the inverse on the bottom. And so, for example, I think, yeah, you put, so for potassium, I think it's outside concentration of potassium will be 5 or 4, and then the inside will be, I think it's 150 in terms of the concentration. But then you go back to the, so its contribution to the resting membrane potential is about minus, I think it's minus 80 millivolts. I can't remember the exact number.
Well, and yeah, this is kind of what I was getting at with the spherical cows. Is you'll read a textbook, it'll give you a value, but a different textbook will give you a different value, and then it's different in different cells. So both in terms of the concentration and the final values. But yes, you kind of, I think, and in terms of this equation, I don't think you're going to have to remember it. I guess there's possibly a small chance you could be asked to explain it if you were given it, or maybe there might be a multiple choice question on it where they might ask, I can't really remember. You'd probably know better than me. But I think it can be helpful to get a grasp on this that can help you explain things. So yes, you talked about yes, the differing effects, potassium and sodium. And yeah, you mentioned there's the difference between chloride. So why is that different?
Is it something to do with valency? I can't remember that. I've gone through it and I've forgotten again.
That's all right. Yeah, no, so yes, you're certainly on the right track there. So there's, I guess, two things you need to look at: the charge on the ion, whether it's an anion or a cation. So chloride is negatively charged, whereas sodium and potassium are positively charged. And so, yeah, I don't really understand all the mathematics of how they derive these equations, but ultimately, in the final form, it results in the negative ions being kind of flipped around compared to the positive ions. So that's why it's the internal concentration of chloride is on the top, where it's the outside concentration for the others two that's on the top. Yeah, the other question is, and I guess we can introduce here, these are obviously not the only three ions that are present in cells or outside cells. We could throw in calcium or magnesium for that matter. And so, yes, how would they be treated in the context of this Goldman equation?
For example, magnesium, or I don't really know what a good example is. I'm just going to say, for example, calcium. All right.
Yep.
Calcium has a valency of 2. It's positively charged, so it's a cation. It has a higher extracellular kind of concentration compared to, I think, intracellularly at resting state. So you've got the two. If I was to put it in the equation, then it'll be like on the outside will be concentration of calcium would be on the top.
Yep.
Then to account for the 2 valency, it'll be at the beginning. So like 2 permeability of calcium bracket calcium on the outside over 2.
Yeah. No, I think that sounds right. So yes, having you just whack a 2 in front of it and just treat it mathematically like that. So that's my understanding as well. So yes, but in terms of, I think the contributions of those other ions to the resting membrane potential are comparatively small. So that's why they don't bother including them in this equation, but you certainly could do that. And at this point, I think I'll see how this works. So there's actually a Nernst-Goldman simulator. Someone's gone to the trouble of putting all these equations into a computer program that you can access. It used to be available at a link where you could go to it online. That's no longer working. There's downloadable versions that you can download, but they require Flash, which is kind of no longer really a thing. So they've stopped working, but you can get it in app stores. So certainly for iOS, you can get it. It's not available on a Mac as yet. So that means I'm going to try and do something a bit different and try and share it via my iPad. Let's see if this works. Okay. Okay. It doesn't seem to be. Let's try that again. So you're not getting anything on your screens there, I'm guessing?
No.
That's right. Let me just managed to get it to work last night, but doesn't want to play. Oh, there we go. Yeah, so this is this calculator, and you can see up the top you can look at just a single ion and go through the Nernst equation for that, or you can combine them all together in the Goldman equation. And so you can see there's the 3 main ions that we're interested in. You can also adjust the temperature because that also has an effect on it. And so you can see here there's different lines across the screen for the different equilibrium potentials for each of the ions as it currently stands, and then that black line going across is the overall resting membrane potential. And so you can see what happens is you change the conductance of the different ions, how that affects the resting membrane potential. You can change the concentration inside and outside the cell and how that affects the equilibrium potential, what happens as you change the temperature. And then you can do it for different cell types as well. So this is the default. There's a generic cell. You can do it for a red cell. And this is where it comes in that, yes, depending on what cell you're talking about, that will affect what values you give. But I think in terms of the examinations, they'll just want something that's vaguely in the ballpark, acknowledging that the different textbooks say different things. And then, yes, they've got the classic squid Axon values there. And so, yeah, if you're kind of a visual learner or you want to play around and kind of maybe try and get a better idea in your head of how these things work, that can be one option. I'm not sure if I think on the blog it's still got a link to the web page that doesn't work anymore. I'll see once I can get access to update that again. I'll add a link into the App Store instead, and the website where the people that developed it came from, because there's also some extra information there where it goes through some of the explanations behind the different things as well. So that could be a helpful resource if you think that will be helpful to you. So now I'll try and get back to our presentation. I might come back to that simulator later on when we get to action potentials. Okay, yep. So any questions at this point about that or anything else before I move on? No. Okay, so this let's try that again. Yeah, so this is again is yes a spherical cow of a typical cell. This is from the Rang and Dale textbook, which is actually a pharmacology textbook, but it fits in well with physiology as well at this cellular level. And so yes, it's got some values here, and I can't remember if Rang and Dale is still one of the recommended texts. I certainly found it useful when I was doing the primary exam. So yeah, these are probably a typical cell, so some ballpark figures that you could use if you wanted to. And yes, it also, this kind of diagram kind of sums it up pretty nicely. It shows some of the different pumps there. It's got the resting membrane channels up here that kind of, they're the ones that are always open and creating that leak that generates the resting membrane potential. You've got the ATPase here. It doesn't have the values of the different ions, but that's what generates the concentration gradients that are so important. And then there's some other kind of ones that aren't really relevant to this discussion, but have some effect there. And so you've got the intracellular, extracellular concentrations, and then the equilibrium potential for each, and then the overall resting membrane potential. So it gets a figure of about 60, but yeah, I think generally in neurons it's considered to be even kind of lower than that. Okay, so next one. So yes, so we've gone through this resting membrane potential. Oh, before we move on from the resting membrane potential, there is on the blog website the link to quite a good little summary paper that explains how the generation of the resting membrane potential, and that's certainly the best resource I found when I was trying to kind of sort it out in my own head. So make sure, yeah, if you want to, if you're struggling with the concept and want to do some reading, I recommend that article on the blog site. But yes, we've got this resting membrane potential, but we talk about muscle and nerves being excitable cells. So what does it mean to be an excitable cell, and how do they get excited? So what's kind of the next step?
So if there's an action, are you talking about if there's an action potential? Yes,
yes. So yeah, tell me a bit about an action potential, and yeah, well, I guess yeah, let's how do we get to an action potential? So let's start off in the cell body, the soma here, which we've got a micrograph of here. What are the events that are going on inside there in order to excite the cell and generate an action potential?
I'm not really sure quite down to cell body level, but there would be, so in terms of potassium going, or sodium going in and potassium going out, and you've got to reach, so it's got to get to a threshold potential. So the membrane potential, say if it's resting at -7 or -80, the sort of in and out flux of the ions will change so that they reach a threshold potential, say around -60 or -50, and then the action potential can propagate. So that's when you get sodium going into the cell, which will be making it more positive, and then sort of going on from there, and calcium as well.
Yep. Okay. So
kind of. So yeah, if we're talking about a nerve, then yeah.
So so you mentioned kind of those ion fluxes with potassium or sodium getting up to that threshold potential, which we'll come to in a little bit. But I guess how do we get to those thresholds? What is it that causes those fluctuations in ions? What are the inputs to this cell body that are causing those changes in ionic concentrations in the cell that gets it to threshold in the first place?
So if an action potential comes from a nearby cell, voltage-gated sodium channels will open. Yep.
So that's so a neural input, and I guess ultimately that's a chemical input. So yes, you've got another neurons very rarely, if ever, exist in isolation. They're more often than not attached to other neurons. So there'll be another neuron that will synapse on it, and it will open some ligand-gated ion channels. So that's kind of a chemical stimulus. What other kind of stimulus to a neuron can you think of in terms of, what are our
You can get a noxious stimulus or a chemical or a mechanical stimulus to trigger a nociceptor.
That's right. So there's all those other kind of sensory inputs that we're used to. They need to be able to be transduced into the cell somehow, and there's still lots of questions about how that works. But yes, things like mechanical forces, so light touch or heavy touch, other chemicals, so as well as neurotransmitters that might be cytokines or icosanoids, those sorts of things will interact at their receptors. Electrical signals, I guess if you get electric shock or something like that, heat, thermal, that there's kind of thermal sensors, and if it gets too hot, then that'll become a pain sensor as well. So there's all these different inputs that will go into the cell, but ultimately that input will be kind of translated or transduced across the membrane as an electrical signal through some kind of signal transduction mechanism. Yes, if you're at my earlier tune on cellular physiology, talk there about lipid rafts in the concept of the mechanism of action of general anesthetics. Well, there's certainly discussion as well about the role of lipid rafts in some of these mechanoreceptors and thermal receptors as well, and disrupting these lipid rafts may be part of the mechanism that results in opening of ion channels and having that signal transduction of these non-electrical inputs. We've got these electrical fluctuations within the cell body from all these different inputs. And yes, I guess one example of that are what's called EPSPs and IPSPs. Has anyone heard these terms before or know what they stand for?
Are they like excitatory postsynaptic potentials and inhibitory postsynaptic potentials?
Yes.
And they relate to the cation or the anion, kind of that they, allow flux through, and hence cause an excitatory or an inhibitory kind of effect to the nerve.
Yeah, that's pretty much it, spot on. So these are a specific example of those different mechanisms that can, I guess, push a cell towards an action potential, or perhaps move it away from it. And so you're right there. An excitatory postsynaptic potential. So this is specifically talking about where there's been some other neuron has a synapse onto the cell body, or the dendrite possibly. So yes, there's different parts of the neuron where you can have these synapses occurring, which are actually relevant, as we'll come to a little bit later on. But yes, depending on what that neuron is that's synapsing on it, it will kind of release an excitatory or an inhibitory neurotransmitter, and then that will have an excitatory or inhibitory effect on the cell. And this is kind of a graphical illustration of that. And so you've got the membrane potential there on the y-axis, and it's kind of not actually at 0. That's just kind of the reference point for this graph, and showing the effect that these excitatory postsynaptic potentials and the inhibitory postsynaptic potentials have in terms of affecting moving away from that resting membrane state. And so these are not action potentials as such. These are just a kind of a local fluctuation in the membrane potential at that synaptic site, the postsynaptic site of that junction. And yeah, so I guess fitting into that excitatory or inhibitory categories, can you think of some specific examples of the different neurotransmitters that might generate either one or other of these responses in the postsynaptic cell? So let's start with excitatory. What are some of the excitatory neurotransmitters?
Glutamate.
Yep, that's one of the big ones, definitely. Any others?
I'm not sure if noradrenaline and acetylcholine in the sympathetic ganglia is relevant or not relevant.
Yep, certainly. Yeah, no, I think that's absolutely relevant, and particularly, so I think acetylcholine has a very big role to play in the central nervous system. It's just that a lot of the drugs that we use that interfere with acetylcholine don't cross the blood-brain barrier, so we don't kind of pay too much attention to them until we start using drugs that do cross that barrier. So yeah, certainly acetylcholine, and then thinking outside the central nervous system, in the peripheral nervous system, autonomic nervous system, that sort of thing. Yes, the noradrenaline and related groups have that, and then there's also, so I think 5-HT3 is an excitatory one, and even ATP itself can have a direct excitatory impact. What about the inhibitory ones? There's a couple of big ones. I'm sure there's more than that, as we'll come to.
Glycine, I think. GABA.
Yep. Yeah, they're the two big ones that I was thinking of. And in the interest of time, so this is another slide from Rang and Dale. And it goes into, yes, quite a few different agents that are either excitatory or inhibitory on cells. And yes, some of these are neurotransmitters, some of them are locally acting, some of them are xenobiotics. And yes, most of them you've never heard of, and they're experimental or historical. Yes, it's just kind of a good summary slide of the some of the agents, and then what ions they kind of act through as well. That just shows that, yes, there's really quite a lot of them once you start digging a bit deeper. Now, so there's kind of a generic term for all these little electrical disturbances, these EPSPs, IPSPs, or the things that are caused by, not by neurotransmitters, but by locally acting or other sensory inputs into the cell. And those little fluctuations in the cell membrane. And so it's this idea of electrotonic potentials. Now, it's probably a foreign term to you, and I don't think you need to necessarily know this stuff for the exam, but hopefully by going through this process of explaining some of these deeper down concepts, it'll help us to explain or help understand some of the more macro phenomena that are examinable. So electrotonic potentials, basically an EPSP and an IPSP are an example of an electrotonic potential. So it kind of refers to the electrotonus, the term, refers to the passive spread of charge inside a neuron and potentially between cardiac muscles or smooth muscle cells, if you kind of might have time to talk about them later. The important thing is that it's kind of the passive spread of charge. So whatever it is that caused the original disturbance, so there's some kind of channel that's been opened, or an experimenter has stuck something into the cell and injected something. So there's been some active disturbance, but then the spread of charge passively within the neuron refers to this electrotonic spread, or the disruption in the membrane potential is an electrotonic potential as opposed to an action potential. So an action potential is active, an electrotonic potential is passive. And it's also dissipative is the word that they use for it. So it kind of dissipates over time and distance. It's not kind of self-propagating like an action potential. It kind of slowly but surely disappears over time. And there's a good video. I'm sure most of you are probably aware of the Khan Academy videos. They have a good description of electrotonic potentials and how they differ from action potentials. And I've got a link to that in the presenter notes in my slides. And I'll also probably stick that on the website when I get a chance as well. So yes, the key things about these electrotonic potentials are that they're passive and they dissipate over time. And so this kind of visualizes it in a sense. So you've got some kind of nerve cell here, and so some initial electrical disturbance, and the trigger could be a neurotransmitter, some kind of cytokine, a sensory receptor, or an experimenter sticking a micropipette into the cell and injecting some sodium ions into the neuron. So that, as you can see down below, disrupts the membrane potential just in that very localized area. And so now you start getting this passive kind of diffusion of the electrical current or the ions. And again, it'll be down their electrochemical gradient, as we talked about with generating the membrane potential in the first place. And so again, they're moving passive, and it kind of dissipates, as you can see. So this is I guess a more theoretical, mathematical, physical representation of what we're talking about here. So kind of a stylized neuron or axon up the top as a cylinder, then you inject current at that point, and then on this case it's distance on the x-axis. So if you measure the voltage as you move away from that point where the injected current was, it decays exponentially in space on this graph, but also in time as well. So if you measure the voltage at that point of injection over time, you'll notice that the voltage, the potential difference there that's been induced will decay exponentially over time. This electrotonic spread, as they call it, is essentially how current is conducted in a wire, except in a metal wire, it's electrons that are moving, whereas in a neuron, it's ions that are moving. So that's electrotonic potentials or spread. But as soon as we start talking about exponential decay here, then that can bring us on to the idea of time constants. So you're probably, depending on where you are in your study regime, you're probably familiar with this concept, but in respiratory physiology rather than neurophysiology. So yeah.
Sorry.
Yeah, go.
Can I ask a question?
Yes, sure.
This might be really stupid, but what's the difference between an electrotonic potential and a mini end plate potential? Are they two different things? Well, I think
I would suspect, I haven't looked at it specifically, but I would suspect we'll get to miniature end plate potentials in a little bit. But I think a miniature end plate potential is an example of an electrotonic potential. It's a subset. So electrotonic potential is a generic term applied to all sorts of electronic disturbances in cells of any kind. And then the miniature end plate potential is a very specific subset of that. Does that make sense?
Thank you.
Yep, okay. And as I said, we'll come to that as we move down the neuron and into the muscle cells. So time constants. Does anyone want to have a crack at explaining what a time constant is?
Annabelle, go. Go around.
No, you're on it, Murray.
Scissors, paper, rock.
Scissors. So time constant, sorry if I get it really wrong, is it the time that it takes for an exponential process to finish if the initial rate of change was to continue, where one time constant is roughly around 63% of the whole process, and around 3 time constant relates to around 95% of completion of the process. And it can be kind of used in respiratory physiology. Time constant equals compliance times resistance.
Oh, perfect. Yeah, couldn't have said it better myself. Yeah, so you pretty much nailed it. So yes, it's mathematically a time constant. It's used to describe the rate of change of an exponential process. And as you said, yes, it's the time at which the process would have been complete had the initial rate of change been allowed to continue. And yes, there's those kind of 3 values that I kind of vaguely remember from my primary exam. I think it's 63%, is it 14%, and then 5% or something after 1, 2, and 3 time constants. Anyway, yes, those specific values aren't relevant necessarily for this talk, but yes, they do seem to ask questions about that, particularly in the context of, as you said, respiratory physiology. And so it's pretty much exactly the same, because we're talking again about the exponential decay of those electrotonic potentials. We can do a analogous thing with the time constant in a neuron. And so yes, it's pretty much the identical formula, and this idea of time constant is relevant in all sorts of kind of physics, engineering, biological sciences, where there's exponential processes going on. There will be some kind of time constant that describes it that more or less matches up with this same kind of formula, because it's kind of describing the same phenomena, it's just in a different system. And so, but yes, because we're now talking about electronics effectively, and they kind of talk about the idea of cable theory. And so a lot of this application to neurons comes from just studying the same things in electrical wise. And I think in particular, when they started laying submarine telegraph cables across oceans, they used these kind of physics and time constants and other aspects to look at the signal decay depending on the features of the telegraph cable that they were laying. And yes, so now they can apply much the same thing, because at its basic level, a neuron has a lot of analogy to a electrical cable in a sense. And so they use these same concepts as well. So this time constant, so for a neuron, instead of being the resistance of the airways, it was kind of talking about the resistance of the neuron membrane is what's the important factor here. And then instead of the compliance of the lung unit or alveoli or whatever level you're talking about, instead of compliance in that physical system, in this physical system, the comparative term is capacitance of the neuronal membrane. Yes, there's this graphic that I've cut and pasted is from a very good website that kind of describes these concepts well. And again, I'll make sure that there's a link available in the tutes to that website. And so, yes, that's kind of the time constant, and we'll kind of flesh this out a little bit as we go along. But as kind of you might have noticed back there in that the slides of the electrotonic spread, I don't know about in lungs, but certainly in cables and neurons, there's a similar concept called the length constant. Now, yes, this will be less familiar, but it's intuitively similar because, as I said, with electrotonic potentials, you get exponential decay, not just over time and the time constant, but it's also over distance as you move away from that initial stimulus. And so you can then develop a comparable thing with a length constant. So it's represented by the symbol lambda, and it's a measure of how far the voltage travels before it decays to zero. So if the length constant is equal to 1 millimeter, that means that at 1 millimeter away from the cell body in an axon, 30% of the voltage magnitude remains. So again, it's those same values of 37%, 14%, and 5%, but it's related to length rather than time. And so, and yes, I've got another. So yes, and so it's a kind of a different formula. So yeah, this one's we're kind of less familiar with because we don't so much talk about length constants in respiratory physiology, but it's some similar things in there. So it's got the resistance of the neuron membrane again as the numerator in that equation, but then the bottom value there is the internal neuron resistance, and so that affects the length constant. And yes, none of this is examinable as far as I'm aware, but again, I'm going through this hoping that it'll help us to get a better understanding and grasp on some features that we kind of do need to know a bit more about. And so I think this graph here kind of demonstrates it quite nicely. So this electrotonic spread and the length constant. So again, you've got a stylized neuron or axon up there. You've got this site of depolarization, whatever might have triggered that in the first place. And then you get this longitudinal spread outward and the movement of charge down there, electrochemical gradients in response to that disturbance of the membrane potential at that place. And as we said before, it will decay exponentially, not only over time, but over distance. And so the graph at the bottom shows this length constant for a couple of different neurons in the blue and the black there. And it shows that, yes, when you get to that, that time constant value, it'll be different. And there's kind of a bit of a hint there as to what things in reality might affect these constants and their relevance to physiology and how we might interact with those cells. So I hope as I said, you don't need to worry too much about that, but if you can grasp those concepts a little bit, hopefully that'll help us in the next few sections, because yes, one area where this does have an influence is this concept of summation. So we're kind of still in the cell body area. And yes, does anyone have seen this word before, know what it means in this context of a cell body?
I don't know if it means that the action potential will be determined by the net sum of the different electrochemical gradients that have flooded in and out. I don't know.
Yeah, no, that you're on the right track there. Yes, so it's kind of one of the clues is in the weather. So sum, it's kind of adding up something. So it's the sum of things. And yes, has anyone else heard this term before? The different types of summation that we're interested in.
Is this where it leads to temporal and spatial?
Yep.
Yes,
yep.
That's right. Ben, do you want to explain what they are?
So yeah, adding up in so it's the sum, and if it's temporal, I think that's related to frequency, and if it's space, it's related to how close the stimulations are physically.
Yep. No, that's pretty good. And so yes, we've got a picture from Ganong here. And so, yeah, I'm pretty sure that Ganong's still on the recommended list of textbooks. And I remember being told, I don't know how true it was, that if there's something that's in, it's kind of the recommend, I think, yeah, what I was told was that all the questions for the exam have to be written with reference to the recommended text. So in a sense, it means if it's stuff that's in a recommended text, it's theoretically examinable. So that's why I've kind of put some stuff in that might be unlikely to be examined, but theoretically could be. And so, yes, this is from Ganong again, and this idea of temporal and spatial summation. So yes, the temporal summation is you've got one neuron impacting on the dendrite of another neuron in that situation A up here. And so yes, you've got two stimuli separated in time. So it's a temporal thing that we're talking about. And if they happen close enough in time, then they can be added together, they can summate. And this is where this issue of the time constants and the length constants come in. So which is kind of why I initially started going through them, because if these electrotonic potentials, depending on their time constant, that will impact whether they summate over time or not. Because you could theoretically have two identical inputs, but because of differences in the cellular environment, that could affect their time constants, and that will then affect how well they're summated or otherwise. And similarly, so for spatial summation, that's where you have two different neurons impacting on one neuron. Now, how close those stimuli are in physical space will determine whether or not they get summated. And again, having a long length constant for the electrotonic potential makes it more likely to get summated, whereas a shorter length constant means that the electrotonic potential will die out or won't go far enough in distance to kind of meet up with the other one. And so this is kind of one area where these length and time constants become relevant. Any questions about that? And yes, does that relationship to the length and time constants make sense?
Yeah.
Okay, how are we going? 4:30. Right. Okay, so yes, this kind of summation, as indicated, is kind of happening in the cell body, and yeah, it's kind of generally considered like a mathematical or algebraic integrator of all these differing inputs and kind of working out some kind of value as to whether or not it needs to respond to all these multiple inputs that it's getting. So yes, we've got all these electrotonic potentials occurring on, in, and around the cell body and the dendrites as well. So yes, it's there's multiple locations where synapses can occur or other inputs can occur. So it can get quite complicated, and they're being algebraically integrated by the cell body. And so what happens next? What's the next step that we're kind of following through with neurons? And where does that next step happen?
Is this referring to like the next step being the all or none response?
Yes. So yes, it's kind of we're getting to the action potential. So yeah, and I can't remember if it was Mari before mentioned the axon helix. So you can see that there on the diagram. And now it's at the very beginning. You can't see much of the axon in this particular diagram, but that's the very beginning of the axon, and it's a particularly sensitive to changes in the membrane potential. There's a lot of sodium channels there, and it's kind of thought to be kind of the point where all this integration of the stimuli come to a head, and then it's at the axon helix where it gets to this threshold potential that we touched on before. So yes, we'll kind of, yeah, just I think, yeah, we kind of talked about threshold potential a bit before. So yeah, kind of what happens at threshold, we've kind of talked about the integration of all these signals, gets it to the threshold potential, and then what happens from there.
When a threshold potential is reached, so that's around -50 or -55 millivolts, depending on the I guess membrane.
Textbook.
They're fast gated sodium, voltage gated sodium channels, which are like very fast. They open up, that causes a massive influx of sodium channels, followed by like a delayed efflux of the potassium channels. But essentially that influx of sodium channels causes the action potential.
Yep. So yes, we'll kind of go into the action potential in a bit more depth. And so, yeah, this is another slide from Ganong, kind of talks about this threshold potential. And so there's different, so yeah, in this example, it's got the resting membrane potential is -70 and the threshold potential about -55. And yeah, each of those orange curves above the x-axis kind of like different fractions of the threshold intensity superimposed on the same timescale. So this kind of shows the electrotonic potentials and how they kind of decay and how long they take to decay. But then yes, if you get this summation of them, getting up to that threshold potential, then you'll get an action potential reached. Now, yes, the concept of the threshold potential is a little bit confusing. I'd always kind of thought of it as like a pretty fixed concept, but and that's probably kind of how it's treated generally. But there's clearly stimuli that are, and again, this is a bit of a spherical cow because it will differ from there's multiple different types of neurons, and for any given neuron, they'll be under different conditions at different times, depending on what the human's doing, what the ambient conditions are like, and other things that have happened beforehand. But yes, there are some stimuli, or these summated electrotonic potentials, that are clearly sub-threshold, that they're never going to trigger an action potential, and there are some that are so big that they're super threshold, super maximal, more than enough. But in between, there's a little bit of a grey area at this threshold potential, where in one instance it might trigger an action potential, whereas in another instance the same level of depolarization won't trigger it. And so there was one thing I read that almost kind of sometimes when they're doing it experimentally, they'll almost have kind of a MAC value for the threshold potential. So they'll consider the threshold potential as the amount of depolarization that will trigger an action potential 50% of the time. I don't know how widespread that is, but I guess the main thing is there are things that are clearly sub-maximal, super-maximal, and then there's a bit of a grey area at the threshold potential itself. But again, yeah, I probably wouldn't have mentioned that, except it's in Ganong that they talk about it. So I thought I'd just kind of throw that in there, but I hope that hasn't confused you unnecessarily. Okay, so let's just see what my next slide is. Yeah, so yeah, let's talk a bit more about the action potential. So what are its characteristics? So before we get to some of the specific cellular mechanisms, if you got asked what are the important features or characteristics of an action potential in an exam question, what are some of the buzzwords or phrases that you would like to get out? Someone earlier mentioned all or none, so that's kind of one of the classic descriptions. So you might want to elaborate on what that means, and there's a few other kind of key words.
I think you'd have to also mention, so depolarization, and then your relative and absolute refractory periods.
Yep.
And then
Yes.
repolarization.
Yep. Yep.
You could also talk about it being unidirectional in relation.
Yeah. Yes. Yes. We'll come to that in a little bit. Yes, there's certainly stuff there. So self-propagating is one. So once it's unlike the electrotonic potentials, which kind of die out eventually, the action potential is self-propagating. So once it's started, it's just going to keep going and propagate itself rather than needing more electrotonic potentials behind it to keep it going. So the all or none phenomena. So that's, yes, you can't have half an action potential. Once an action potential triggered, it'll be a full action potential. And so under, again, these kind of artificial, all things being equal, fixed conditions, the action potential will have a constant amplitude and velocity down a given neuron. You won't have half a action potential or a double action potential. And it's an active conduction, but it's, even though there are some analogies to a metal wire, it's slower compared to a metal wire because it's ionic movement rather than electron movement, and in terms of the conductivity of the things that we're talking about. The other key thing is that it's an example of positive feedback. So kind of crops up a number of times in the primary syllabus where you want to talk about that. You could theoretically get asked a question, give me some examples of the different feedback mechanisms in the human or whatever, and that's where you can talk about positive and negative feedback, and this is a classic example of a positive feedback situation. So you get that a little bit of sodium influx, and that promotes more sodium influx, so it continues accelerating, so you get this massive spike in the depolarization. Yeah, so I'm talking about the analogies with a wire, which kind of break down at some level. So the alternative analogy that I've heard is talking about a trail of gunpowder. So you've seen in movies and cartoons where they'll be laying down a trail of gunpowder, and then someone will light a match at the end and it will start burning down this trail. Now it's relatively slow moving, but you can't, no matter what you do, you can't really put it out by just treading on it. But if you wanted to, you just need to make a break in the line of gunpowder and it'll fizzle out. And that you can kind of, that's effectively what we do with local anesthetics along neurons. We kind of break that chain of gunpowder so that the action potentials aren't conducted. So yeah, if someone wants, yeah, we've kind of started talking a little bit about here, so I may as well go on to the next slide now. So the ionic changes here. So does someone want to go through these steps here? So I've got 1, 2, 3, 4, 5, 6, 7. So what's happening at step 1 on the green curve up the top?
So that's I believe that's the resting membrane potential.
Yep. And it's important to remember that when nothing's happening, a lot's happening. But yes, so that's resting membrane potential. Then on to 2. What have we got there?
So that I thought was summation of graded potentials up to threshold.
Actually, yes, I've just realized that those numbers, there's the next slide, and I think this is an animation in the digital Ganon version. So those numbers correspond to states of the sodium and potassium channels that are involved. So yes, maybe we can frame it in that. So at that first one there, what state are the sodium and potassium channels in?
I believe they are both closed.
Yes, it probably depends on which potassium channels we're talking about. There's the potassium leak channels that'll be open, but then there's some kind of other active ones as well. Okay, so what about at 2? What happens at 2?
So the fast voltage gated sodium channels open, I believe.
Yep, sounds about right. We'll check it on the next slide. Oh, your guess is good, yes, but it sounds right to me. What about 3?
Then I thought the slower voltage gated potassium channels start to open.
I think that's probably right.
Anyone else, please correct me if I'm wrong. And then at 4, the sodium channels close.
Yep.
I'm not sure what else has changed at 5.
Yeah, actually, that doesn't. Oh, they might. Yes, they might. Let's just go to the next slide. So we can. So I think there's probably some other text that I haven't got on this screenshot that describes what those numbers mean. But I think some of them relate to what's happening to the channel. Some of them relate to what's happening to the relative membrane permeability in the graph below it. So again, yes, this is possibly a decent, again, it's from Ganong. So it's one to come back to, I guess, correlate the membrane potential to the different conductances that is happening, and then the underlying changes in the channels that are causing that. Because yes, as you kind of mentioned, yeah, the differences in the sodium channels and how they open and close in the different states they're in, which will become relevant in the next section. Okay. Yes, so I might, any questions at this point? Does that graph make sense? Yep. Okay, so yes, given that we've said, yeah, we've talked about the all or none principle, that once the threshold is reached, the action potentials are more or less identical, irrespective of what the magnitude of the initial stimulus was. So how then is increased stimulus intensity conveyed to the target organs or downstream tissues? How can the brain convey that, the level of intensity of the input, given that the action potential can't change its nature?
Is it frequency?
Yep, so that's, yep, one option. So you can have a given neuron firing more frequently, and that will provide repetitive, that kind of temporal summation of the signal, I guess, downstream to either the next neuron or the muscle cell or whatever the end organ is. So yes, so increasing the frequency of action potentials kind of increases the information or the stimulus. So what's the other option? Talking about, that's an example, I guess, analogous to temporal summation. What would be the spatial summation equivalent of increasing the stimulus intensity?
I know in terms of pain, you can get recruitment of C fibers that otherwise wouldn't be involved in transmitting pain.
Yep. No, so I think that's, yeah, so you can have more neurons firing. So you can have, get an increased stimulus transmission through recruiting more neurons. And another example of that is motor units, which I don't know if we'll get to that later on, but yeah, so you might have one motor neuron, might innervate a fixed number of myocytes, but then you can recruit additional motor neurons to recruit additional muscle cells if you want to increase the force of that muscle contraction. So yes, you can increase the frequency of neuronal firing and the number of neurons firing to increase the signal intensity, given that you can't increase the intensity of the action potentials. Okay. So yeah, we've kind of touched on this before. So what is it that stops an action potential going backwards? So you talked about it being unidirectional, I think someone did. So yeah, what stops it going backwards?
The refractory period.
Yep. So do you want to, actually, before we get on to that. So yeah, certainly, yeah, the refractory period, you kind of get that period where the bit behind the wave of action potential can't be stimulated. But it is, in some unusual circumstances, or possible to get an action potential going backwards in some sense. So I guess, yeah, classic example is if along the length of a neuron, there's some stimulus there, either artificial or experimental or just accidental, you will get the action potential going in both directions from that disturbance. And I even think, I'm not sure about this, but I certainly remember hearing that the reason whacking your funny bone feels funny is because you're kind of hitting your sensory neurons there, and you're actually getting an action potential going in the wrong direction, down your sensory neuron towards your hand. And that is an example of what they call antidromic conduction of an action potential. So dromic kind of means a course, or a race course or a road, a bit like a velodrome. And so orthodromic conduction means conduction of the action potential in the traditional or normal direction, whereas antidromic is in the opposite direction. And so, yes, that's while we do talk about it being unidirectional, there are some examples where it can go backwards. And sometimes I'll even talk about, so we talked about the axon hillock that triggers the action potential. I have seen some discussion where, as it triggers the action potential to go down the axon, it also sends the equivalent of an action potential back into the soma, back into the cell body of the neuron, to kind of reset that whole thing to zero effectively. And so then it can start from scratch, interpreting the new incoming signals. But that's, I think, yes, very much fine print and not really examinable. But yes, the refractory period, started talking about that. So let's go on with that. So who wants to expand a bit more on what the refractory period is or means, and I guess what underlies it?
So the refractory period is the period of time where the nerve, and can be also used in pacemaker cells, can't depolarize. Mm-hmm. You can split it into absolute and relative. Mm-hmm. And it's a period of time where I'm not sure exactly what causes it, but it's, the cell is repolarized and can't depolarize again. I'm not sure if it's because the ion channels are closed. Mm-hmm. I'm not sure.
Can you expand more on the absolute versus relative concepts there?
So the absolute refractory period means that no matter the stimulus, that the cell can't be depolarized to propagate another action potential. Whereas the relative refractory period means that if the stimulus is large enough that it can overcome that ability to depolarize again.
Yep, no, that all sounds right to me. Does anyone want to have a crack at describing the cellular mechanisms for the refractory period?
My understanding is that the sodium channels can only reset if they become sufficiently repolarized. I don't know if that threshold's like -30 or not. So if they're, as long as they're above that limit, then they can't be re-stimulated. They're not open and ready to go.
Yes, so I think, the membrane potential and its value probably has some role, and then there's also, I think, a time role as well. Does anyone else have any other thoughts on the underlying? I'm not 100% certain on this myself.
I know that the existence of the calcium channels that keep it more positive for longer in cardiac cells means you've got a big plateau, whereas in neurons you don't have those calcium, so it repolarizes quickly and you've got a short refractory period.
Yeah, that's right. So we probably won't go too much into the cardiac cellular physiology here. We'll go into skeletal muscle a bit more, but there'll probably be other tutes on the cardiac side of things. But yes, certainly calcium plays a big role there. But I think there's also this aspect, I think it was in one of the, so yeah, this one there's, so the sodium channels that are involved have these different states of inactivity. So I can't remember the exact terms they use to describe it, where I think it's the different, like to be able to be activated again, these sodium channels have to go through a number of different states. And there's one of those states where they just can't be reopened no matter what. When there's a subsequent state where they prefer not to open, they're not quite back to normal, but they will open if you push them hard enough. And I think it's, yes, there's some gating mechanism there where it's kind of physically blocked for a bit, where, yeah, nothing will open it. You might want to kind of double check all this stuff. But yeah, I think there's probably a role there for the just the timing mechanism of the sodium channel, and that'll probably differ from different neurons, and probably also between neurons and skeletal muscle and other tissues. But I think there's, yeah, as well as the repolarization that's necessary, there's also just some kind of fixed time in terms of the nature of the channels, the sodium channels themselves. Okay. Now this is another slide from Ganong that I found confusing, but it's in Ganong, so hopefully this won't confuse you more. But these graphs kind of show the membrane potential change kind of over time, and then also kind of correlate it with the excitability of the cell. So how easy it is to excite the cell, to excite the neuron in terms of being able to get another action potential out of it. And the thing that I found confusing was that it kind of talked about this refractory period down in the excitability graph at the bottom, but then it had this supernormal period, which I couldn't understand how that related to the absolute and relative refractory periods. But I'll come to that in a little bit. So yeah, what it's kind of showing here is that with that initial, as it's kind of developing, getting towards, you're getting this local response, the electrotonic summation at the axon hillock. In that phase, as it's kind of summating and getting closer to the threshold, it actually becomes even more excitable at that point. So there's almost this non-linear response even early on, where as you get a little bit of depolarization, that makes it more sensitive to additional depolarization. So in the build up to an action potential, you're getting this period where it's really quite excitable. And then you get the action potential, and then it becomes completely unexcitable at that point. And now I think, yeah, where I was misunderstanding things was that this refractory period referred to here includes both the absolute and the relative refractory period combined together. And then I eventually managed to chase down the reference this graph was from, and they talk about in some cells, not all neurons, some neurons, after that period of refractoriness, there will be this supernormal period, this after depolarization, where it's actually more excitable than the normal baseline level of excitability. A brief window when it's actually easier to excite than normal. And then following on from that, there will be this longer after hyperpolarization, where it's less excitable than the normal baseline. So again, I think this is kind of, it's in Ganong, so that's why I've included it, but it's a little bit different to what you might read in other sources. And I think part of the problem is that this is describing the situation in a particular subset of neurons, and it's not necessarily generalizable to all of them. But yeah, the main thing to point out is that when you dig into the data, this refractory period covers both the absolute and relative, and then in some neurons, there might be a period after the refractory period where it's hyperexcitable for a little bit, and then less excitable for a longer period after that. And that might be due to prolonged opening of potassium channels or something like that that trigger that. Okay. Any more questions about that before we move on? Or anything else? Okay, so yeah, so we've got the action potential, and there's yes, different axons that the action potential can go down now. But yeah, as we've already touched on before, there's these myelinated axons. So what can you tell me about myelinated axons? What are its characteristics? What do we know about it? What's it made of?
I don't know whether I know much about it, but essentially my understanding is that it's made from those oligodendrocytes or Schwann cells, depending on where they are, and they insulate the axons. They provide insulation in regular intervals, and hence concentrate the voltage-gated sodium channels in regular at the nodes of Ranvier. By that, provides saltatory kind of, I don't know how to pronounce that, but conduction.
Yep, that sums it up pretty well. So do you know which types of neurons you tend to find myelin around?
Anything that needs fast conduction, whatever that is.
Yeah, so that's an important, yes, you get the faster conduction with myelin. So I think, yeah, what I've read anyway, but I think there's subtypes within each of these. So I think A and B will tend to have myelin, whereas the C fibers tend not to. Then it gets a bit complicated because you'll sometimes have multiple unmyelinated neurons encapsulated in a outer shell of myelin to varying degrees. So it does get a bit complicated. And so yes, you get that fast conduction. They look different, so they tend to be white rather than grey. Have I got that the right way around? I think. And yes, it's an insulator. It's do you know what it's made of? Anyone? What's its kind of chemical properties or biological properties?
I just know that it's there's 100 layers.
Right, yeah. Yes, so yes, it kind of wraps around multiple times, doesn't it? And so it's pretty much just the whole cell, aside from its kind of nucleus, is pretty much just lipid. It's I think it's particularly sphingolipids, which is a particular type of kind of cell membrane type thing. So yes, it's just kind of lots and lots of the cell membrane, and I think it's got a bit of cholesterol in it and stuff. But yes, it's basically this very thick, multi-layered lipid. And yes, kind of going back to the cellular physiology tutes, we know that in terms of electrical movement across cell membranes, lipids are very resistant to that. So they make a very good electrical insulator, which is its key role. And you also mentioned there, Marie, I think it was, the oligodendrocytes versus the Schwann cells and central versus peripheral nervous system. Is there any kind of clinical relevance to that differentiation that anyone knows of? Do you know of any neurological conditions that affect myelin?
Is it anything to do with MS?
Yes.
Or-
So what can you tell me about MS? What does it affect?
Not much, but it's a autoimmune disease characterized by like a sort of attack and degeneration of the myelin sheaths. I'm not sure if it's of peripheral myelin.
Do you want another guess?
Central.
Yes. So MS is central demyelinating disease. Can you think of a kind of a analogous peripheral one?
Guillain-Barré.
Yeah, that's right. And so I suspect or assume, I haven't actually looked into it, but it wouldn't surprise me that if whatever the immune mechanism is, the MS one is directed towards oligodendrocytes, and the Guillain-Barré one is directed towards Schwann cells. And that kind of explains the differentiation between central nervous system and peripheral nervous system. But that's just my theory. So, and I don't think they'd ask that in an exam. So not in the primary exam. Yes, and that's a bit of a niche meme there. You need an interest both in neuroscience and Australian broadband infrastructure. That's the node of Ronvier, just down the street from me. That's where I get my NBN from. So yes, Louis Ronvier was the one that described the nodes of Ronvier that we've been talking about. So yes, kind of myelin is a lipid-rich cholesterol and some proteins, but yes, this sphingolipids. Talked about Schwann cells, talked about oligodendrocytes, talked about that. And then, yeah, someone's already mentioned this. I think this movie's getting pretty old now, so probably won't get the reference, might need a new meme for next year, but yes, I have trouble pronouncing the word as well, but does anyone know what saltatory means? What the word means? Something to do with table salt or not? I think it's from Latin, like a lot of this stuff is. So saltare apparently means to hop, leap, or dance, is what I've been told. So that's this, yes, kind of the idea of the action potential kind of hopping down the neuron rather than just kind of being kind of progressing down in just a more straightforward fashion without the myelin. And so, yeah, this is where kind of the previous discussion of these electrotonic potentials and the different constants kind of come into play. So this is, yeah, kind of the diagram that I had up before. And so as we talked about, the electrotonic potential decays exponentially as you move away from the source. So this move away in length as well as time. And yeah, and part of that reason for that decay is you get current leakage across the membrane. So yeah, we know that there's the membrane is leaky to potassium in particular, even under resting conditions, and then other channels can potentially open depending on the level of the disturbance. So when you get this site of depolarization, for whatever reason, you'll get some leakage, as indicated there, back across the membrane. Now, as we've said, myelin kind of insulates that the neuron. So you get slower decay, and that's kind of indicated by this blue line here. You still get the exponential decay, but the length constant that we were talking about before is much longer than in a smaller unmyelinated neuron. Yeah, so we can see here, yeah, so going back to first principles and these, the length and time constants, mostly interested in the length constant here. So what effect will myelin have on the length constant, given what we've just been discussing?
Would it reduce the resistance of the neuron membrane? So does it? I don't know.
So what have we described myelin as? What kind of properties does it have, electrically speaking?
Insulation.
Yeah, so if you insulate something electrically, does that increase or reduce the resistance to current flowing across it? Increase. Yeah, so myelin will increase the resistance of the neuron membrane. So then what effect will that have on the length constant, according to that formula up the top there?
Increase it.
Yes, that's right. And I think it'll probably be on the next slide, actually, we'll go into it. But yeah, on the denominator on that equation there, so the internal neuron resistance, what things do you think might affect that? So this is talking about the resistance of current moving inside the neuron rather than across the membrane. Given we kind of talk about this as cable theory, what do you know about electrical cables and things that affect the resistance of current moving through an electrical cable? I'm sure most of you probably know the answer to this, but you may not have thought about it in this concept, but this kind of explains one of the, maybe I'll reframe it. So what are the things that speed up, make conduction velocity in a nerve faster? So having fast nerve conduction is important for some nerves. What are the characteristics of those nerves that have the very fast conduction velocity?
So myelinated, large diameter, high concentration of voltage-gated channels at the nodes of Ranvier.
Yep. So yes, tell me about the diameter. So why is the diameter important?
So it'd just be the similar principle to an electrical cable. So a larger diameter, you can have more flow of, is an electrical cable, large diameter, you can have more flow of electrons.
Yep. Yeah, and so that's exactly what's happening with that internal neuron resistance. As the diameter of the neuron increases, you reduce the resistance to that electrotonic spread. And so as you reduce the resistance inside the neuron to that current flow, then that also increases the length constant. So a larger diameter neuron wrapped in myelin, you can dramatically increase the length constant of the electrotonic spread in that neuron. The time constant gets a little bit more complicated because there's opposite effects of myelin. And given that none of this is really examinable anyway, we won't go into that. But just I think the next slide will kind of show the importance of how this length constant and the factors that affect it, how they affect the conduction of neurons. So this is again from that same website that has those formulas on it. So this shows a neuron without myelin up the top compared to one with myelin down the bottom. And so you can see that without the myelin, so the graph, so this kind of voltage, kind of showing the voltage inside the neuron. And so this is the electrotonic spread or electrotonic potentials that we're talking about. And you can see compared to the one without myelin to the one with myelin, the exponential decay is much quicker. So it means that you need to have these voltage-gated ion channels kind of much more frequently, and each activation of these voltage-gated channels takes time. So you want to have as few of those activations necessary as possible. And this is where the role of myelin comes in. So once you've got good insulation, plus or minus a larger diameter, the rate of decay of the electrotonic spread is much slower. So it lasts not only much slower, but it goes much further down. The length constant is much longer, so that electrotonic spread will reach the next node of Ronvier. And as you've also said, there's a much higher concentration of voltage-gated channels there, so they're much more likely to be triggered. They're much more sensitive, so even a smaller ion voltage change will trigger their opening. So you get that triple effect there. So that means that a single electrotonic potential will reach further down the neuron, and it'll reach a spot where it's really kind of ramped up, ready to go. And so that means that if you compare those two, the one up the top with the one down the bottom with the myelin, there's only been 2 necessary activations of those sodium channels, where up the top you needed 1, 2, 3, 4, 5. And if each of those activations takes a finite time, then you're going to dramatically increase the velocity of spread in the myelinated nerve, particularly if you add in not just the myelin, but the larger diameter as well, because that will make that length constant even bigger, so you can have the nodes of Ronvier even further apart, if you want to. So does that make sense, given, yes, kind of introduce some unusual, unfamiliar, non-examinable concepts, but kind of the goal of that was to try and help you get a better understanding of how the myelin actually works at kind of the molecular electrotonic level. Any questions about that before I move on? Should probably get going, given we've still got the muscle cell to go. And I'm happy to take questions at the end or by email or whatever. And yeah, that's just going back to that original slide that we had. And yes, in terms of talked about the location of the sodium channels that are voltage gated. So I think, yeah, that's right. They talk about the myelin and those faster nerves. They can be 50 times faster with myelin. So it's quite a dramatic increase in conduction velocity. And the number of sodium channels per square micrometer of membrane. So in myelinated mammalian neurons, it's been estimated as so 50 to 75 per square micrometer in the cell body. At the initial segment, the axon hillock, that really sensitive bit to what's going on in the cell body, it's 350 to 500 per square micrometer in that initial segment. On the surface of the myelin, it's less than 25 per square micrometer, but then the nodes of Ronvier, it's up 2,000 to 12,000 per square micrometer. So there's a much higher concentration of sodium channels at those nodes of Ronvier compared to anywhere else in the neurons, which is important. Okay, neuromuscular junction. Who wants to have a crack at describing its features, or what are the important aspects that we need to know about? This is something we're intimately interested in as anaesthetists, so we kind of need to know a fair bit about it. What can you tell me about it?
Okay, so we've got the, in terms of neuromuscular junction, my understanding is that you've got the synaptic, like it's the, am I getting it
right?
Yep. I'm just getting a brain freeze as I speak, so just give me.
That's all right.
Someone can go if you if they need to talk. But neuromuscular junction, is that the area where you've got your areas of troughs and crests? No, hold on. Is that not? Am I getting it wrong? Am I?
So what's the name suggest? So we've got neuro. What's that referring to?
Yeah, it's like the area where well, your junction of your nerve meets motor.
Yes, that's right. So you're so you've got a motor neuron that then synapses on a muscle cell, and there's a synapse there. So can you anyone tell me more about that that synapse and kind of some of its physical or chemical features?
Yeah, I'll have a stab.
Go for it.
So the neuromuscular junction, so you break it up. So there's the presynaptic motor end plate. And
is that presynaptic?
Oh, there we go. So no, that's postsynaptic.
Yep.
So you have the postsynaptic motor end plate, and then do you still call it the terminal bouton?
Yes, I like to use the French whenever possible. Yes, I think that applies to any synapse from the neuronal side, but yes, it's often used in the context of the neuromuscular junction.
Yep. So I draw the terminal bouton as like just a normal sort of synapse, and then like Mario was saying before, the motor end plate, you draw it with little peaks and troughs.
Mm-hmm.
It contains vesicles, so there's vesicles that are stored way up in the synapse. Sorry about that. And then there's vesicles that are stored closer to the membrane.
Mm-hmm.
And then I'd also chuck in on the motor end plate, there's the receptors, so the acetylcholine receptors.
Mm-hmm.
And then in the troughs, you have the acetylcholine esterase waiting to sort of mop it up.
Yep, that sounds pretty good. Let's see. So you mentioned the acetylcholine receptors. Can you tell me any more about them?
So they're a pentameric receptor.
Mm-hmm.
So in the adult, there's 2 alpha, 1 beta, 1 delta, 1 gamma. No, 1 epsilon, sorry.
I'll trust you on that one.
Yeah.
What's the subtype of, talking about acetylcholine.
Oh, yep. So they're a ligand-gated channel.
Yes.
And they conduct...
So we'll get to that in a little bit. What can you tell me about acetylcholine receptors more broadly?
Yep. So acetylcholine receptors get activated when you have binding of acetylcholine on two of the receptor binding sites.
Yes. Sorry, this is a bit of a game of guess what I'm thinking. So there's what subtypes of acetylcholine receptor do you know of?
Oh, I don't know. Oh, nicotinic muscarinic. Yeah.
Yes, there you go. Sorry, you won the game of guess what I'm thinking. Yes, I think we've got most of it. We'll just go. Actually, yeah, so let's talk about, just quickly, some drugs that act at the neuromuscular junction. There's a few that we're particularly interested in.
Yes, so you have your neuromuscular blocking drugs that act at the nicotinic acetylcholine receptors.
Yep.
And then the ones that work at the muscarinic receptors.
Well, let's keep it to the neuromuscular junction.
Yep.
So so you talked about the neuromuscular blockers.
Yep.
What other kind of drugs? And I guess, you could break that down into depolarizing and non-depolarizing. And there's a kind of at least one other class of drug that we use regularly that acts in and around the neuromuscular junction as well.
So there would be things that operate in that space as well, like neostigmine or idrophonium, but they mainly work via inactivating acetylcholine esterase, I thought.
Yeah, but that's still in the neuromuscular junction. So kind of the neuromuscular junction is taking as a whole the kind of the motor neuron synapse, the terminal bouton, the synaptic cleft, and then the motor end plate, and kind of all the reactions that take into account of that. So trying to think of it as a whole integrated unit. What about Sagamodex? Does that work there?
Not as far as I'm aware. So Sgamidex is the large molecule that exists in the plasma, and I'm fairly certain that its mechanism of action is that it captures the rock uranium molecules, and then the capturing of that rock uranium molecules forces a concentration gradient back from the NMJ back into the plasma.
Yep. Does it work with any other neuromuscular blocking agents?
Works with pancuranium and antibiotics and oral contraceptive.
All right. There's another neuromuscular blocker as well that it.
Pancuranium, becuranium.
Yeah, Beck. Yeah. But yeah, and yeah, it's the oral contraceptive is an important one. I think we're meant to give them advice, mind you. Yes, I'm not too sure what would be going on straight after surgery that might require that for 24 hours, but it's an important medical legal thing in particular. And yeah, it came as quite a surprise to me. It was well after I'd been using it quite a bit that I was aware of that issue with oral contraceptives. There's another one that we don't so much use, but surgeons sometimes use, or certain kind of subtypes of surgeons use that actually it's a bit more left field. And given the kind of the time, I won't kind of drag the chain too much. So Botox, botulinum toxin, it works at the neuromuscular junction. So the Clostridium family, of which Botox is part of, is a bit of a mongrel of a family. We'll get to tetanus in a little bit, but yeah, Botox prevents the release of acetylcholine from the neuromuscular junction, so it causes that flaccid paralysis. So yes, in terms of the pharmacology of the neuromuscular junction, actually, I think there's another slide coming up that will go into that a bit more. So yes, this is a stylized diagram of what we've just been talking about. I think we've covered most of the aspects there. Okay, we'll skip through that. Yeah, so this is again from Rang and Dale, so kind of the pharmacology side of things. So yes, it's covers a lot of those things that we've already talked about, the different agents, and yet again, some of them we still use regularly, not so much the tubocurarine these days. There's other examples of non-depolarizing blocking agents that would be more useful, but I suspect they haven't updated this diagram since it was first done many years ago. But then there's also some historical and experimental drugs that are used there that work in different ways. Mentions the botulinum toxin there. And then there's also some presynaptic receptors that are kind of probably relevant. And then also go back up further from my cellular physiology tutor, acetylcholine comes in. Again, it's kind of a central mechanism in cellular physiology. Anticholinesterase like neostigmine. So yeah, that's kind of, it's a bit messy to begin with, but could be a good reference document to come back to if you want to kind of revise the neuromuscular junction pharmacology and as how it fits into the physiology. So yes, this is looking in a bit more kind of closer detail. And so maybe we can go through these numbered steps here. So let's, yes, I don't know if you want to tag team or if someone wants to have a crack at all 7. Let's start off with number 1 up the top there. So what is this number referring to? What's going on here?
The action potential.
Yep, so you've got the motor neuron, action potential coming down. And what's happening at number 2 here?
That's the presynaptic voltage-gated calcium channel being open to.
Yes. So yeah, that's a voltage-gated. You get a bit of calcium influx. Number 3, what's that?
That's the exocytosis of the acetylcholine vesicles.
Yep, good. Number 4 here
is the binding of the 2-acetylcholine to the ligand-gated nicotinic acetylcholine receptor.
Yes, well done. Okay, number 5 here.
That's the influx of the cations, usually the sodium, that's causing the opening of the voltage-gated sodium channels.
Yeah. So actually, yeah. So what can you tell me about this process here? So what's happening in this ligand-gated ion channel?
So the ligand-gated ion channel is so it opens it opens when there is 2-acetylcholine, as then described.
Yep.
And when it does open, it's usually, it's not necessarily cation specific, although any cation can technically go through, although it's most sensitive or most conducts lets sodium pass.
Yep. What's the other ion of interest that can go through?
Calcium.
I don't know. There's another big one that's clinically relevant. So we've got a cation channel that's open. What are the important cations that we're interested in terms of membrane potentials and influx and efflux?
Is that what you're referring
to? So I've got sodium going in. Is there something that could come out that wants to go down its electrochemical gradient?
Yeah, potassium.
Potassium, yeah. So you can get sodium going in, but you can also get potassium going out the other way. And that's, I think that's the presumed mechanism as to why after giving succinylcholine, you can get an increase in plasma potassium levels because it kind of leaks out through those acetylcholine receptors. That's the explanation I've heard anyway. I'm not sure if where I actually read that. But yes, it's important to realise that yes, unlike a lot of the ion channels that we're talking about, this one is not as specific as others. So you can get, and you may well get calcium going through as well. I'm not sure. So yes, number 5, you started talking about there. So yes, just go back over that bit again.
Yep, so that's the activation of the voltage-gated sodium channels.
Yep, and so this here is kind of another example of that electrotonic spread or electrotonic potentials that we're talking about. So this is a passive process. So we've got an active process here that causes this localized depolarization. And then it's a question of whether that can spread far enough in time and space to then get to the voltage-gated sodium channel. So yes, number 6.
Is the sodium maybe flowing through?
Yes. Yeah, I think so. And then, and yeah, actually, so while we're at this point, so something that I kind of kept getting confused with, and other people I've spoken to did as well, that we can often miss out this step number 5 conceptually. Like we just think that once acetylcholine binds, we open the this ligand-gated ion channel, and then sodium kind of rushes in and depolarizes the whole muscle cell. But you can't forget this important step if you get this localized depolarization here, and it has to electrotonically spread to the nearby voltage-gated sodium channels to open them. It's not these voltage-gated sodium channels that are opened by the acetylcholine directly. There's this intermediary step in number 5. And then finally, number 7. So what's happening with number 7?
Yeah, that's the spread of the action potential.
Yes, exactly. Yeah, no, I had exactly the same. Like it took me a while to realize that it's not only neurons that have action potentials, but muscle cells do as well. And so yes, the skeletal muscle very much has an action potential, and it's pretty much an identical mechanism to the neuron in terms of these voltage-gated ion channels and kind of self-propagating across the muscle cell. And yeah, so yeah, any questions about those steps before we move on? Clarification or extra points people would like to add in? We'll kind of talk about something a bit more specific. So yes, I think earlier someone mentioned miniature end plate potential. Yeah, so does anyone know what a end plate potential and a miniature end plate potential are and what the difference is between them?
Sorry, once again, I don't know whether this is completely right. So my understanding is that a miniature end plate potential is when the that is something that happens kind of quite spontaneously and doesn't necessarily reach threshold, but it's this, not, I guess, the summation, I guess, of all the miniature end plate potential plus whatever that happens that causes an end plate potential. Well, if it reaches threshold. I don't know.
You're definitely on the right track. No, yeah. So we'll go back a little bit to the previous slide. So the end plate potential just refers to kind of what's going on here when acetylcholine binds. So you get the opening of these ligand-gated ion channels, and yes, you get this influx of sodium, and so you get a depolarization, and that's called the end plate potential. Now, normally, because it so rapidly then goes on to produce the action potential, when you're measuring it electrophysiologically in a lab, you normally don't actually see this end plate potential because it just gets swamped by the much larger action potential. But there's various drugs and mechanisms you can use to kind of hide the action potential and still measure this end plate potential. So this, the end plate potential is just describing this localized depolarization that then undergoes this electrotonic. So it's, I guess, yeah, the end plate potential is an electrotonic potential that then, if it's large enough, will then spread to the voltage-gated ion channels and cause an action potential. Now, a miniature end plate potential is a kind of a specific description. It's kind of another type of electrotonic potential, but it's what happens out. So you mentioned this kind of random release of vesicles. So because the neuromuscular function has to be so rapid and reliable, all the acetylcholine vesicles are really primed up and ready to release their acetylcholine at the drop of a hat, at the slightest whiff of calcium. And there's kind of interesting molecular mechanisms as to how that happens. But what that means is that occasionally, even without any calcium influx, it'll just accidentally release a vesicle of acetylcholine into the synapse. And that's what triggers this miniature end plate potential. So it's a very small response, again, difficult to measure. But I think this is technically in the syllabus, or was when I went through, so that's why I've covered it here. And so this kind of shows the difference between them, and you can see the difference in the x-axis here. So this is talking about the resting membrane potential of around kind of 76 or so. So these miniature end plate potentials are these tiny little spikes here, and they represent individual quanta of acetylcholine vesicles being released and maybe binding to some acetylcholine receptors and just triggering a very small miniature end plate potential, whereas this is kind of the summation of a action potential coming through the neuron, triggering the release of a whole lot of acetylcholine into the synapse, and then what the motor end plate voltage reaction to that is when it gets that. So that's just, yes, a distinction between end plate potential and the miniature end plate potential. So they're kind of related because they're both at the end plate, but they're kind of, one is the spontaneous accidental release of acetylcholine, whereas the end plate potential is kind of the intentional release of a whole stack of acetylcholine. Phase 1 versus phase 2 block, I don't know if that's still a thing. We don't give many succinylcholine infusions these days. Given the time, might kind of skip over that one. Except, yeah, the main thing to, so yeah, the phase 2 block is when you had repeated or prolonged doses of succinylcholine. And going back here, it results in desensitization of these voltage-gated ion channels. So it almost induces a longer refractory period in for the action potential. Whereas I think early on I was confused that the phase 2 block was because you desensitize these the acetylcholine receptor themselves. but phase 2 block is related to desensitization of the voltage-gated ion channels. And that'll probably do that. That's a pretty minor print thing these days. So yes, skeletal muscle action potential. Obviously, yeah, it's after 5:30 now, so if people need to go, that's fine. You can catch up on the recording later, but I'm happy to keep going if people are happy to keep listening. So the skeletal muscle action potential. So yeah, as I said before, it's pretty much identical in the underlying physiology in terms of how it's transduced. But it's important to, yeah, remember, we don't just lump straight from the neuromuscular junction to muscle contraction. There's this intermediate step of the skeletal muscle action potential. And in relation to that, what can you tell me about the structure and function of the skeletal muscle fiber and how it relates to its action potential? What's your understanding of the way the muscle fibers are set out?
So the structure of the muscle fiber has these, which is different to like say smooth muscle, is that it has these regular T tubules or invaginations that cause rapid, I guess, depolarization of the whole muscle fiber.
Yep, that'll do as a starting point. I've got a diagram again from Ganong here. So yes, you've got these little invaginations. So the sarcolemma is just a fancy name for the plasma membrane of the muscle fiber. And you've got these kind of little holes here that kind of form these T-tubules. They don't really, the transverse tubules, yeah. So these are the T-tubules, so those invaginations that are kind of go in and wrap around. So this is the T-tubules are basically just, topologically speaking, continuation of the plasma membrane of the muscle cells. So the action potential, once it starts propagating away from the neuromuscular junction, spreads not just along the surface, but goes right in and around all these, the different filaments. And you kind of going from the macroscopic right down to the microscopic and down to the cellular level, which we'll get to in a second. But yes, the important thing is these transverse tubules, and then we'll come to the sarcoplasmic reticulum in the terminal system in a little bit. So excitation, contraction, coupling, is that a familiar term? Does anyone know what that means or what it's kind of describing? Or does anyone want to have a crack at describing how it happens with the skeletal muscle cell?
Yep, I can give it a crack.
Yep.
So it describes the interaction between the like, I guess it describes the process of from the action potential arriving at the myofibril and having a and it's, I guess, interaction between the release of calcium, sarcoplasmic reticulum, as well as the contraction coupling between the actin and the myosin, leading to the sliding filament theory. So I guess in terms of the process, once an action potential really reaches the through to the T tubule, you've got the L-type calcium channels being opened, which causes calcium to be influxed in. That causes the activation of the ryanodine kind of 1 receptors to be activated at the sarcoplasmic reticulum, which causes a massive calcium surge in the cytoplasm from up to like 10 micromoles or whatever. I don't know how much it is. That the calcium then is able to bind to troponin C, which then causes a conformational different change to the actin, and causes 2 things. It slides the tropomyosin off to expose the actin binding site, as well as it also causes the troponin I, or the inhibitory troponin, to allow the myosin ATPase to kind of react. The myosin ATPase, so myosin has, which is part of the thick filament, has, it's initially bound to ATP, but when it degrades to ADP and phosphate, it's able to latch onto actin, which causes a power stroke. And I guess that's what.
Yes, that was very thorough. I think you've covered about the next 10 slides. No, that's very good. I'll go to, I think the next one's got, yes, so yeah, as I said, I think you've covered pretty much everything. There's just, you mentioned, yeah, so you've got the action potential coming along the sarcolemma and down into the T-tubule. And then you've got the, yeah, these are the L-type or dihydropyridine receptor channels here. Now, you mentioned that they open and let calcium in. So does anyone know of a distinction between, so these L-type channels are present both in skeletal and cardiac muscle. And there's a subtle but important difference between those found in skeletal muscle and cardiac muscle. Does anyone know what that is and I guess the relevance of it? It's a bit of game of guess what I'm thinking, but. No, it's fine. Yep. Interest of time, I'll crack on. So, yeah, Murray, you mentioned that yes, these are a voltage-gated ion channel, calcium channel, and yes, they do open and let calcium in. But the key thing is that in skeletal muscle, the influx of calcium through the L-type channels is not actually required to activate the ryanodine receptors. There seems to be some voltage-gated mechanism. So the voltage change from the action potential results in a conformational change in the L-type channel. And that conformational change is enough to activate the ryanodine receptor. Whereas in cardiac muscle, you do actually need that calcium influx through that channel to then stimulate the ryanodine receptor to open and have the calcium efflux from the sarcoplasmic reticulum. And so that means that it's a much faster coupling mechanism in a skeletal muscle than in cardiac muscle. So again, that just exemplifies every step of the way with skeletal muscle activation is ramped up for speed. So from the large diameter myelinated neurons to the neuromuscular junction with the ready to go primed acetylcholine receptors. And also we didn't kind of cover it there, but they release way more acetylcholine than is needed. And then you get these invagination, the T-tubules, so that the action potential goes everywhere in the muscle cell really quickly. And then even down to this molecular mechanism here, it's a voltage mechanism rather than a calcium-induced calcium release mechanism, which it is in the cardiac cell and in other cells. So that's just a subtle but important difference in that L-type calcium channel there. And then pretty much everything else you said was pretty much right. So I'll go through the slides here. So yes, yeah, you described here this is a cross section of the thin filament here. You've got the troponin, the actin, the tropomyosin. This is in the resting state. You've got the ADP here and the myosin kind of bound. And then when the next slide, calcium comes in, binds to the thin filament and the troponin C, I think it is, that exposes, oh yeah, no, yeah, so this is where you get the exposed binding site and the myosin can now bind to the tropomyosin. Yes, I'm struggling to remember all this myself now. And then in the next one, you get the power stroke. So it's kind of ADP here. And during that power stroke, it releases the ADP. You get the longitudinal force that's generated at that point of the power stroke. And then at the end of the power stroke, ATP binds to this exposed site. And that causes a detachment from the actin filament, and then goes back to the resting state as that ATP is hydrolyzed, and you get the ADP there, and the phosphate is released at that point, and that kind of recocks the myosin head, so to speak. So yes, I think you covered that pretty well. But then the next question is, so how is contraction switched off? And so we've kind of gone through how the action potential and the transduction of that signal starts contraction. So how do we switch it off when we need to?
Is it to do with the intracellular calcium concentration?
Yep.
So the calcium can undergo several mechanisms for the concentration to go down. One is its active transport back. No, is it active? I don't know.
Yeah.
Transport back into the circuit by the circuit kind of transported back into the sarcoplasmic reticulum, as well as the calcium-sodium antiporter at the sarcolemma kind of area, as well as calcium ATPase at the, again, sarcolemma as well.
Right. Yep. No, that sounds right. Yeah. So the circuit that you're referring to, that's just short for sarcoendoplasmic reticulum calcium ATPase. So yes, that's definitely an active transport mechanism because it's there's a lot of calcium in the sarcoplasmic reticulum, and so it's pushing it up a concentration gradient. So that's ATP dependent. And then yes, there's some other mechanisms, but I think it's mostly that reabsorption into the sarcoendoplasmic reticulum, so it's ready to go for the next contraction when it's called on. So the next question is, what happens if we run out of ATP? Going back to those previous slides.
So running out of ATP would kind of cause that rigor mortis kind of where it just can't latch off.
Yeah, that's right. So we've got, we kind of go back to the, so it's kind of done that longitudinal force, the ADP is released. Now, in order for it to relax again, you need to put ATP back into the myosin head. And so if you die, you're no longer producing ATP. And then also as you die, you kind of, everything else starts to fall apart. And so there's release of calcium into the cytosol of the muscle cells. So there's plenty of calcium around. So it will contract, but there's no ATP to cause the relaxation. So you get that rigor mortis until other degradation processes take over. Oh yes, and I guess this is where it's probably relevant talking about malignant hypothermia. That's obviously something you need to know about. Yeah, do anyone want to have a quick crack at what they know about malignant hypothermia? How would you describe it? What's the relevant things we need to know about it?
Well, it's a rare but life-threatening.
Yep.
A life-threatening condition triggered by certain agents like volatile anesthetic and succinylcholine that cause an unregulated change in calcium homeostasis in the skeletal muscle, where there's a surge of calcium. Intracellular calcium causing symptoms of XYZ.
Yep. We won't go into. Yes, that's obviously kind of. I think you can could get us this stuff in the primary, not just the final exam. And so one of the buzzwords that I was taught to use if I got asked anything about it was it's a pharmacogenetic disorder that emphasises that there's both a genetic component and a pharmacological component. So it's not just those drugs that trigger it. You need to have the underlying susceptibility to it as well. And pharmacogenetic is a quick way to describe that when you're trying to save time. And yes, calcium homeostasis is another good buzzword to use. And do you know where the genetic aspect of it comes from? What's the protein that's affected mostly? Yes.
It's an autosomal dominant condition that happens with the mutation of the ryanodine 1 receptor.
Yep. And I guess, yes, that kind of talked a bit about the L-type calcium channels and how it's different in the skeletal versus cardiac muscle. And I think that's the same with the ryanodine receptor as well. So yes, I'm pretty sure that MH doesn't affect cardiac muscle, and it's a different mechanism there anyway. So yes, you kind of mentioned, the kind of symptoms relating to it. And so obviously hyperthermia is one of them, along with all the other issues. So given what you've described as to what it causes in the cell, what is it that actually causes the symptoms that we see? What are the, what's the cellular mechanisms there? So you said it's related to calcium. So how does that cause the further symptoms that we see?
So the rapid the massive surge in calcium causes increased force of contraction, which causes which you see with the masseter spasm, or like the tensed masseter and the muscle contraction. And that muscle contraction can, and it can also that can also lead to increased heat. I think, is it because of the rapid ATP that you're using, or is it because, I don't know, why do you get hypothermic from it? Is it because you're like contracting more? I don't know, sorry.
No, you're kind of on the right track. And so yes, we are isothermic, or I can't remember the exact term, but like we can maintain our body heat, so we do need to produce heat as part of our normal metabolic process, and I kind of cover that a bit more in one of my tuts later in the year. But so the hypothermia, asking where this heat comes from. So any metabolic process is not 100% perfect. There will always be some energy lost as heat, and that's what maintains our body temperature normally. But here it's getting out of hand. And the question is, so that's generally from burning ATP, and you're right. So the muscle contraction, so you can get that muscle rigidity, but that will only use up a certain amount of ATP. But probably the main source of ATP consumption is we talked about wanting to turn the muscle contraction off by pumping calcium back into the sarcoplasmic reticulum, and that's an ATPase dependent process as well. But the problem here is that because of the defective ranitidine receptor, any calcium that gets pumped in just gets re-released back into the sarcoplasmic reticulum. So you've got the circuit pump going absolute kind of ballistic, kind of going overtime, pumping calcium and using ATP, but it's a futile cycle. It's not actually reducing the concentration of calcium inside the cell. So yes, you will get some increased muscle rigidity, but not always. That can be pretty subtle, or it might only be temporary. But yes, most of the subsequent symptoms are from the increased metabolic needs generated by the calcium pump. So you get the hypothermia, and then once you get the hypothermia and the other issues, you start to get cell breakdown, and so that's when you get lysis of the muscle cells, and you get increased potassium release and myoglobin anemia and those sorts of things. And so, but yes, the kind of the root cause, the issue is mostly the calcium pump, but also a bit of the ATP used for muscle contraction. But that's comparatively minor, I guess. And what's the treatment for it? I guess there's several, but the mainstay of treatment, the thing that was kind of a game changer in terms of survivability,
is dantrolene.
Dantrolene, yep. And what's its mechanism of action?
It acts as a antagonist at the ranitidine receptor, and hence causes the circuit, allows the circuit pump to just.
Yeah, that sounds about right. Okay. This is just so just talking about the action potential in the different. Motor neuron, skeletal muscle, and the cardiac ventricle. As I said, we're not really covering cardiac physiology this time, because that should be covered in other tutes, but just comparing the different natures of the action potential. So you can see the motor neuron and skeletal muscle are pretty similar, but the cardiac one is very different. And then, so does anyone know what this painting is depicting? and we're thinking
Tetanus.
Tetanus, yeah. So this is, I think it's, yes, I don't know when the painting's from, but yes, it's meant to be a depiction of someone that's got tetanus and got the muscle rigidity that can be kind of not just lockjaw, as it's sometimes described, can affect the whole body. And so yes, Clostridium tetani is another dodgy member of the Clostridium family. And actually, yeah, so probably shouldn't go into the details now, but the toxin for that actually enters at the neuromuscular junction, but then it kind of moves back up so the neuromuscular junction enters the axon, the motor neuron axon, but then it moves up the motor axon, gets endocytosed, moves up the motor axon, gets exocytosed into the synapse at the top of the motor neuron, then gets endocytosed into the central nervous system. So it actually works at the central nervous system in the inhibitory interneurons, which is kind of vaguely relevant to the exam. And so then it inhibits the inhibition of the alpha motor neurons. So the alpha motor neurons are overactive because they're not being inhibited back in the central nervous system in the spinal cord. Anyway, that's just a bit of an interesting side note. Yes, this is a way of introduction into the concept of tetany or tetanic contractions, because this is something else we can potentially get examined by. So yeah, do you understand, or have you heard of the concept of tetanic contractions of skeletal muscle and what they are? So this is in the normal healthy situation, not in someone that's got tetanus.
Is it the summation of individual muscle twitches and that can cause essentially continuous contraction? I mean, you've got complete and incomplete tetanus. Yep.
Yep, so this is, yeah, you definitely got it there, but what about the refractory period? Why can we get these tetanic contractions when you've got the refractory period of the action potential that shouldn't allow another action potential to develop straight away? Bit of a trick question.
Is it because the action potential duration is so short, and the refractory period actually finishes before the excitation?
Yeah. Yes. No. Yeah. I can definitely. Yes, you're definitely on the right track. And this next graph, I think, kind of shows what you were describing there. So this is another one from Ganong. And so yes, the top one shows the muscle fiber action potential, and the bottom graph shows the tension, the muscle fiber tension. And so yes, you can see the action potential is very short, as we saw before, and it's basically done and dusted, including its refractory period, before the muscle twitch has even gotten going. So it's easy to imagine a second action potential coming along before that muscle's even reached its peak twitch height. So that's why, yeah, so the refractory period is only to do with the action potential. The contraction mechanism doesn't have a refractory period. So it can be repeatedly stimulated with that calcium mechanism we described before in the face of multiple action potentials coming down a single motor neuron. Yeah, so that's just the text describing that. And then, yeah, this is what you were describing there, where you can get just the individual twitches as the frequency of the stimulus increases with those orange arrows. You just get a single twitch, and then you get that summation, similar to how you can get it with the neurons, and then incomplete tetanus, and then you get that kind of flatter, complete tetanus where you just get this prolonged singular contraction. And this is kind of what we're talking about, is that example of given that the action potential is identical on the motor neuron every time, you can increase signal intensity by increasing the frequency of those action potentials. And then that can result in the tetanic contraction. And most of our everyday muscle contractions are actually tetanic. If you're trying to lift the weights or do anything useful, you don't want this varying kind of muscle twitch. But then again, you can recruit additional motor units if you want to lift a stronger weight, but each motor unit is pretty much always working at a tetanic contraction level. And then a cardiac version of tetany, well, not really. We don't really want our heart to go into a tetanic contraction, but I guess the analogous situation is what they call the TREP effect, or the staircase effect, or the Bowditch effect, which is assuming the guy that invented it. And it's basically where and this is where, yeah, the action potential of the in the cardiac muscle is different, and it has a much longer refractory period, so you can't get that tetanic contraction, but it still seems to be this increasing calcium concentration inside the cardiac cells. And this graph, again, I know this is from somewhere else, but it's showing how with isolated cardiac muscle, as you increase the frequency of contractions, the contraction force of the muscle fiber increases. So I think this is where it's going 20 seconds between contractions and then less than a second between contractions. And you can see the force kind of starts to increase and then reaches a plateau. And then it kind of slowly kind of drops off as you go back to that 1 every 20 seconds. And this is a different kind of experiment where you're going from a stimulus every 0.2 seconds and a very high level of force developed to 1 every 300 seconds, and the force has dropped way off. And so it's the same muscle fiber, everything else being equal. The only thing that's been changed is the frequency of stimulation. And so that's where, as you get tachycardic, you can get that increased contractile force. Oh yes, and this was, this is again from Rangadale. This is a smooth muscle cell, so it's again different to skeletal and cardiac muscle, and it's very different in terms of its contractile mechanisms. And I don't think I've ever seen I don't think you need to know too much about the specifics of excitation, contraction, coupling, and that. But certainly, yeah, you do need to know about some stuff about smooth muscle. It has important roles in a number of places. And I don't know if this has ever been an exam question, but it's conceivable that it could be. Compare and contrast skeletal, cardiac, and smooth muscle mechanisms. Could be a short answer question. And this out of Rangadale is a pretty good, I think would be a good framework for comparing and contrasting those things. And also this highlights the different signal transduction mechanism for the L-type calcium channels between skeletal muscle over here and cardiac muscle here. In skeletal muscle, it's just this voltage-gated thing. There's no need for that calcium influx, even though there probably is some. Whereas in the cardiac muscle, you do need that. It's a calcium-induced calcium release there. So it kind of demonstrates that nicely. And then smooth muscle is completely different again. So yes, if you want to try and preempt an exam question, that Rangadale picture is not a bad one to do it. Oh yeah, the cardiac muscle, as I said, we won't go into that much today. I think there's in those learning objectives from the primary syllabus, when I did it, had something about the knee jerk and talking about the role of inhibitory interneurons. And this is what the tetanus toxin targets, the inhibitory interneuron here. In some, it may not be this exact one, but it's back in the central nervous system in the spinal cord. That plays a role there. And I think they also talk about in that syllabus page, presynaptic inhibition versus postsynaptic inhibition. So this black neuron here and the yellow neuron are both inhibitory neurons. And so here, this inhibitory neuron is directly inhibiting the motor neuron via this postsynaptic inhibition, because there's this synapse here, and it's after this synapse that you get the inhibition. Whereas this presynaptic inhibition is, so the purple nerve is a stimulatory to the motor neuron, but by having this inhibitory neuron inhibiting the stimulatory neuron, you get a net inhibitory effect. And this again comes back to all that summation and the electrotonic things and how that relates to those excitatory and inhibitory postsynaptic potentials. Yes, I don't know if they have asked about the different skeletal muscle fiber types. That might be in the syllabus again. Oh yes, and this is just going back. So yeah, there's obviously things in here that I haven't covered at all today. Some of them are more important than others. You need to make your own judgment call on that. So yes, I haven't really talked about the autonomic nervous system, which is obviously a big thing we do need to know about. And then that's the muscle physiology as well. Again, some stuff that's, I think we covered most of that in terms of the big stuff anyway. That's pretty much it for me today. Happy to take questions now, or if there's any in the chat that I haven't seen, or yes, it's getting late, so you're more than welcome to nick off now, or send me an email down the track. That it's from me. Any questions?